Okay, I'm live, and hopefully I'll be seeing Aaron the Grower soon. Today's topic is about redox reactions in, I guess you could say, plant health generally. But specifically, we're going to be looking at plants, insect, and other sorts of organisms that utilize redox reactions in, I guess, in their normal metabolic processes. I do feel like some people get very excited about this topic, and a lot of people are very aware of maybe like a particularly narrow sort of example of redox reactions. And I myself am not like a chemist by any stretch of the imagination. I know a little bit of organic chemistry and some inorganic chemistry, and I know a bunch of like really high level stuff that's really irrelevant to just about everything to do with plant health. Um, I think I even a little bit away from me for a example here, but I guess I could bring it up. Um, I have a Maximet knife, for example. I'm a big fan. What's up, Phytoalchemist? What's up, Mobile J IPM Goat? I appreciate that you think I'm goated in the IPM space. I will definitely be saving this live for later viewing, Mama CMO. Yeah, Phytoalchemist, you were right. Redox is a deep and complex rabbit hole. That is definitely true. Oh, okay, here we go. Sorry. All right, he's here.
Oh, great. Can you hear me?
I can. Can you see me?
Oh, I love that shirt, dude. You're the man. You know what's funny is I got Brandon's shirt on.
Well, there we go. There we go. You can see that we're just shills for our friends. Don't listen to anything we say.
Yeah,
definitely not impure. Yeah.
Yeah, it's all incestual over here. That's all we do.
That's right.
So what's going on, dude? Nice to see you.
Yeah, I know. I was actually taking a look at my IGTV because I've been uploading some to YouTube lately. I've been playing around with Premiere Pro, which I recently got. I'm probably not going to use all of its intricacies, at least not recently, but I found out that the last time we spoke was like December of 2020, I think.
I know.
Yeah.
It's been a crazy year, but we don't have to spend too much time on that. I want to get straight to it for people because this is a encyclopedia of information. I mean, beginning this research was like, I have a really nice expression for stuff like this. How do you eat an elephant? One bite at a time. And that's how this research went. It was like day after day, I just spent reading papers trying to get a hold of what this is. And I have a feeling a lot of growers probably had the same experience when they were trying to take a bite out of this. So I want to start really basic and work our way up, if that's okay with you.
That is definitely okay with me because I'm certainly not an expert on this topic.
Oh God, and I'm going to be a lot less of an expert. So, and I got really stoned this morning because it's 7:10. So happy 7:10, by the way.
That's right. That's right. This is anyone, no matter what date it is, when they watch this 7:10 video, I think it's illegal to not celebrate in the way that you subscribe.
I agree. Now, hopefully, I'm somewhat cognizant today, so I'm going to do my best. This is a complicated subject. So, what is redox? Let's jump right in.
So basically, there are a couple of different factors, right? So I know that many people are aware of, and even when you ask that question, I immediately think of the really common example of like valent electron gain and loss, right? So, you gain an electron. Well, actually, back me up here because I'm going to say this the very fundamental thing, and I might get laughed at here. But if
oil rig. Oh, Fido just said it. Oil rig.
Oil rig. Oh, there we go. Oil rig. So
oxygenation is loss, and reduction is gain.
Reduction is gain, right? It's sort of counterintuitive, right? Even I make that mistake often.
I had to run oil rig down in my head so many times that I ended up making it a part of the graphic because it's such an integral part. Like you're saying, it's fundamental, and a lot of this stuff is going to just be really fundamental building blocks before we get into the really cool like inner gut of the insect stuff.
Yeah. Oh, and I just wanted to say, just for my own credibility, I guess, that there's other aspects of this. So we're going to talk about how pH affects redox potential, for example. So environmental conditions, it's more complex than simply like gaining and losing electrons. We've also got protons or hydrogen atoms too. It's a big component as well, which is part of the reason why the pH is important, right?
Right.
So it's not just electron gain and loss. There's other factors to consider, which is why it's so freaking complex.
Dude. And you know, the other reason is it's because there's so many reactions that are actually considered redox reactions. Like when you start diving into this stuff, it's like, oh my God, every single thing in the world starts with redox almost. It's like the mother of all reactions.
In a lot of ways it does. In fact, you know, I have this little reference for me here because I wanted to make sure I said it sort of eloquently. But you know, like basically fundamental cell respiration, at least if we're talking about, you know, aerobic organisms, and even in the case of like animals, we can sometimes in very specific narrow situations, like with mussels, we can use anaerobic. I'm sure you're aware anaerobic respiration happens too, but aerobic respiration that's sort of one of the fundamental paradoxes of life on Earth, is that so many things rely on oxygen, yet oxygen is super reactive. And so you're always playing this game from like basically basically every cell process has to balance like oxidative stressors and how to excrete that and waste removal from those metabolic processes. And also utilizing some of that for defense too. And it's really interesting how these sort of like, in some cases, like waste, you know, metabolic waste refuses get used in a defensive capacity or something like that.
Absolutely. And yeah, Fido, we're going to get into the ROS, the reactive oxygen species. Yeah. So we're going to get into that too. Hopefully, if anybody has anything, I know that there's going to be some people in the comments section here that are going to have really, you know, succinct contributions. So if you feel like you have something to add, please do. We're definitely we're going to try and stay open on this conversation a lot. So now that we've talked a little bit about like redox, is there anything else you wanted to get into about the general sort of stance of redox, how we're going to be approaching it today?
Let me just check my notes here for a moment.
Please
I'll start with a small definition that that I kind of wrote here, just for people who might not be familiar. So I wrote that various agents like chemicals and enzymatic proteins can have, for example, antioxidant effects, right? So it's not just chemicals, right? We're very familiar with like sort of hydroxyl. I guess that's part of the ROS, but like superoxides and peroxides and other sorts of things, chemicals like that, they can have, of course, an oxidating effect. And that's like what we think. I think that's what a lot of people think of. But, you know, various proteins, various enzymes have antioxidant effects, for example. And we'll get into antioxidant effects too. But I think it's important to consider because that's how, you know, insects, for example, are able to, you know, sort of neutralize certain defenses that rely on oxidation. And one of the ways to do that was with gut, but I'm going ahead of myself. I want to say various agents like chemicals and enzymatic proteins have antioxidant effects that neutralize other agents like radicals, which is what I was talking about earlier, things that your body has to deal with. And there's various ways too. There's actually other ways that they can maybe create barriers and other sorts of things to sort of sequester. Maybe that's not the right term. Maybe in this biological case it might be confusing. But in other words, to keep these reactive agents from messing with DNA and other sorts of processes, interrupting the natural flow of these effects. So like a lack of paired electrons is one case. Interacting with hydrogen and oxygen typically makes them highly reactive. Certain radicals are important for metabolic regulation and signaling in animals and plants, while in other contexts they can interrupt these processes and cause problems. Like most biological agents, the effects of oxidants and antioxidants is highly context dependent. And I think that's super important to keep in mind as we move forward, is that your cells and various other processes, whether you're animal or a plant, There's really a lot of things going on, and there's no way that we're going to talk about every single, detail. Partly because the scientific community does not know all of the various factors, but there are tons of people looking into it, of course.
Very well said. And yeah, and let me just emphasize that, as my name implies, I am the grower, not the electrochemist. So this is I definitely want to take like a humble approach to this, and I don't claim that any of this is like set in stone. I'm just doing my best here to you're you're going to guide this conversation with your understanding, and I'm going to do my best to sort of parallel this in an understandable fashion. Not that you oftentimes are perfectly understandable, but
not always.
Not always.
Certainly not this topic. Certainly not this topic.
Hard. This is so hard. So I'm going to just do my best to try and a lot of people are familiar with redox in fire and redox in batteries and stuff like that. Mm-hmm. And even in photosynthesis, that can be a simpler redox reaction. So we'll try and parallel those as we go. But you got into antioxidants there for a second, and that's really something I wanted to understand. I couldn't, again, it's hard to gather from the papers exactly what an antioxidant does, how it sort of serves a purpose within the insects or the plants, generally. How is it produced, how is it used, etc. Please.
So there's a lot of, the real answer to that, as I understand, is that there's tons of ways that they're produced, and there's also various ways that they have a function. And so for example, and just to get to your point about experts and all that stuff, I try and like we do all the time when we talk about these topics on Livestream, I'm drawing from empirical research. And
right.
When you say it's not set in stone, I think that's really important. Even scientific research can be overturned, things can be old, our understanding changes with new information. So just because I say it or that I'm citing research here doesn't mean that it's the be-all, end-all, but it does give at least a little bit more credibility than just some sort of a claim out of nowhere. And certainly on a topic like this, I think it's, like I keep reiterating, it's very complex, and things can be different with various contexts. But here's an example. So let's say that a caterpillar is eating a leaf of some plant, and so as it physically cuts into the tissues, right, with its mandibles, it breaks chunks of the plant off, and immediately puts those into its foregut.
Foregut, right.
So insect gut is super varied between species and between families and between orders. So it's kind of hard to generalize, but I'm going to do my best. I'm going to do exactly what I just said not to do.
And let me just say, before you go on there, a lot of the research focuses around moths, caterpillars, grasshoppers, and then, kind of select other groups. But generally, it's grasshoppers and moths.
There a lot of the research we found, we're at least going to share, definitely concerns that. We have one on color potato beetle.
Oh, aphids. Aphids also. Yeah.
Yeah, and so and caterpillar, first of all, larval gut and adult gut is going to be different. One of the things that some entomologists and other sort of evo-devo researchers have said that I think makes a lot of sense is that one of the reasons why insects are even so successful in the first place is because, at least in the case of the Holometabola, that changed from larva to pupa to adult. So caterpillars to moths, ant larvae to adults, things like that. Something that has a larva form and something that has a very different adult form, like beetles and things like that.
Sure.
So that gut changes a lot, right? And the food that they eat changes, which means that the adults and the immatures don't compete for resources, and so that's really helpful for them. And then they grow fast, they eat lots, especially in the case of caterpillars. And so if there are negative effects, they're able to kind of outgrow them before it becomes a problem. Again, we must consider things like fitness. Just because something is poisonous like just like with us, like something can be very harmful for our health, and it might be acutely harmful and kill us immediately or kill us very quickly. Other things are going to be much more subtle, and they might show their effects when we get much older, when our physiology changes quite a bit, just like in plants and other organisms. So the similar kind of thing happens. As long as the fitness is good and the thing reproduces, and passes on its genes, that's good enough. But to give you an example, let's say this caterpillar is eating the leaf, and the tissues get into its foregut. So insects tend to have a foregut, midgut, and a hindgut, and a lot of insects, not all, have something called a paratrophic matrix, which is kind of like a sort of a web, kind of like a net that only allows like certain compounds through, certain small molecules through. Of course, it doesn't get everything. Pathogens sometimes get through. And certain compounds, and also certain like reactive oxygen species and phenolics and other sorts of things like this, they can damage this matrix, or they can damage the gut, and cause sepsis and other sorts of things like that. Or they can damage the lining and make it so that other microbes that might be taken in with the organism, might be able to actually infect it or do things like this. So in this way, this is where it becomes super complex, right? So maybe the ROS by itself isn't good enough, but the ROS plus a symbiont in the plant, maybe is good enough, right? To hurt that plant, to hurt that organism, hurt that caterpillar, do whatever it's going to do. So there are multiple layers, of course, that we have to acknowledge. But in this like really oversimplified example, one of the things that, like for example, this caterpillar might have a very reduced gut, and I don't mean physically reduced, although that is possible, but I mean from a pH perspective. If the pH is low or high or whatever, this is going to change the redox reaction. And so in a lot of cases, in addition to things like enzymatic antioxidants, chemical antioxidants, and other metabolic processes, redox reactions that the plant might have that would normally damage the insect are going to be totally neutralized, because the environment in which the reaction is going to happen, or going to continue to happen, as it sort of, this is a technical term, but like as it fizzes, as the oxidation starts to kind of happen, as soon as it pushes that into its foregut, if it's a low pH, then those things neutralize because of the nature of pH and redox reaction, or redox potential. And so like when people say
real quick, when you say neutralize, are you saying they become oxygenated or reduced, or is that sort of not a term of redox at all?
I say that like I'm actually kind of blanking on the exact process, and this is part of the me not being an expert thing.
It's part of the pH though, right?
Yeah, well, like
okay.
like I said earlier, so like kind of like, so how pH kind of affects redox, because like you have like hydrogen atoms and things like that affect redox reactions sort of as an environmental effect. So like certain reactions are going to be harder to occur, or they might not be able to occur because they can't catalyze, essentially. That might not be the right chemistry term there.
No, that makes good sense though. Maybe not to a chemist, but logically I understand the semantics there.
So like I do have an example here, I think, that I wrote down specifically for this kind of question, because I wanted to make sure I got it right for people. And I have here written that pH influences redox reactions and can increase or decrease the absorption of nutrients, which is one reason is a factor in, like, for example, the rhizosphere for nutrient uptake of plants. But for similar reasons, gut pH of an herbivorous insect radically modifies the effect of typically defensive redox reactions that might physically harm, deny nutritional absorption. So that's the other thing. So certain nutrients, and don't ask me all of them because I don't know, but certain nutrients are going, and I think specifically like metal ions, so like your sodium, for example, and other sorts of an iron and things like that, those nutrients are going to be less absorbed if the state of the redox is different, or if the state of the gut is different, or if you have antioxidants going in or oxidants going in, that's going to change the nature of the absorption in the gut. And this is true for other animals too. So like kind of goes back to what I was saying, whether it's kind of like a nominally defensive effect, or whether you're kind of denying, like this isn't like an intellectual pursuit on the plant. I guess maybe that's a little bit, I don't know how you want to put it, but like,
I understand, yeah.
right, like essentially
This is like a biological innate approach, but we're kind of approaching it from like a human perspective.
Exactly.
Yeah.
So that's just one example. Also, different sections.
Before we move on, I think and what you're kind of getting into there is the redox potential of the soil. So like, right, like is that like measured in millivolts? And this stuff is all very interesting to me as like an apprenticing electrician, like understanding. Okay, real quick. As an apprenticing electrician, you are studying electrical theory. That is how electrons move from one place to another. Really simply. Well, this is that on like a molecular level. It's and people literally make batteries out of this concept from a molecular stage or scale, and they scale it up to a full-size battery, and you have a redox reaction in mass, and you're having this, so like for example, in a zinc copper battery, you're losing zinc ions from I believe that would be the cathode, and then you're gaining, on the other side, the copper ions in the fluid are oxidizing and attaching to the copper anode. And so without, getting too much into that, that's essentially, if you understand how a battery works, redox is exactly the same thing on a molecular level without humans complicating the process. If I can break that down, that would be the best way.
Well, to bring that metaphor further into something that I think other people might have experience with, in addition, some people might be aware that if you're this is a PSA, I guess, for those who don't know, but if you're charging your phone or another, appliance or whatever, some sort of thing that needs electricity to run, like your computer, if you charge with a charge, like, so sometimes you can get charging cables, and right now I've actually just recently installed 2 USB-C ports on my computer because I deal with a lot of data, and I just want to make that quicker. I don't want to go in megabits. I want to get in gigabits of transfer speed.
I think this is is this USB-C? Is that what this is?
I think so. Yeah.
Like,
is it the same both ways? That's
yeah.
180.
Yeah. Okay.
So
that's the fast one. Yeah.
Yeah, and you'll notice, and USB-C in particular is very, very infamously known for being. There's some that use data, some that can transfer data, some can only transfer electrical current. Some are better at it, some are worse at it, and some things need more or less. And if you don't charge with the appropriate amount of voltage, I believe,
yep,
you will harm the battery. You will harm that sort of reaction that you're talking about.
That's right.
Right?
Yeah, and so you can actually, there's a whole bunch of ways you can destroy a battery, right? But, primarily it's, same thing happens in nature. When you stop that redox when you make that thing incapable of redox, it's dead. It's dying. In a nutshell.
Right, and it's sort of, and like, different batteries have different chemical, constituents, right? And some of them are better for some applications over others. Part of it being with reusability and charging. Other of them, with regards to like how much power can you output.
Yep. Yeah.
I don't know all those intricacies personally, but like.
Yeah, like drones, for instance, they're made with these batteries that take like hours to charge, but they're 15-minute discharge because they have this huge output.
Mm-hmm.
But then you have batteries for your phone that are meant to last, 12, 24 hours, and they take a couple hours to charge, but they don't have a high output.
Right. Imagine they don't last very long, too, in comparison. I guess it depends.
It depends. Yeah, it totally depends on what the battery is made of and how well you treat it and voltage that's applied.
And it's also
Let's.
true that batteries are always dying, right? Don't We should talk more about plant health, but
No, I know.
that
Wait,
is
what'd you say? I missed it. What'd you say?
well, plants. Oh, not plants, but like batteries, chemical batteries. Basically, I know that like they're always like kind of dying in a way, like whether you use them or whether you don't, whether you store them at full capacity or you store them at half or lower. I know that when you transport or when you store them, it's very common practice. I'm not a prepper or anything, but I've looked into some of this stuff for like generators and things like that, or like batteries, and I know that like you shouldn't be storing these things at high. And but anyways, that's not plant health.
Yeah, we'll
But
get back to it. Sorry, Fido. Sorry, guys.
yeah,
That was totally my fault because I actually did want to cover batteries a little bit because it's such a basic way to cover redox. And, we won't get into fire, but fire is also a really basic redox reaction where carbon gains oxygen atoms.
Boro makes a great point. All organisms are always dying
to from the moment you're born, baby.
Right.
All right, so do you feel like, all right, so the purpose of antioxidants then, we covered a lot of that. Is there anything else you wanted to say about that? There will be more of it as we go through, but
yeah, I think that we could move to some examples of things. I know that so I did mention in my post about this that I'll be coming in with empirical data, empirical research, and so I do have this article here I showed you earlier. It's I posted about it too. It's called Redox Control of Aphid Resistance Through Altered Cell Wall Composition and Nutritional Quality. And in it, they take a look at a tobacco plant. They took a tobacco plant that had, and they altered some of them to make a high ascorbate oxidase or have low ascorbate oxidase production in the plant cells. And it's important because what they found was that when the cells were in a reduced state, when the apoplast in particular of the plant cell was reduced instead of oxidized, it was more resistant to the aphid. And also, when the aphid fed on the plant, the sugar content actually increased, not decreased, which is significant because anyone who knows aphids knows that that's basically all they eat. The other thing that, but they need the phloem, they need the sugars in the phloem, but they also need amino acids and things in the phloem as well. Xylem, for those just as basic plant physiology for people who don't know, phloem sap has all the assimilated sugars from photosynthesis. Xylem has all the water channels, and that's for evapotranspiration and that kind of thing. So like some insects feed on mostly phloem, like aphids, I should say, the sucking mouthpart insects, and some of them, like leafhoppers, mostly feed on xylem, and that's very nutrient poor. So they have to drink basically a ton more, as opposed to like aphids, and sometimes they switch. But it's significant because the researchers felt that, not felt, it's a little bit stronger than that. What they found when they took a look at this is that the actual physical construction of the plant cell walls changed because of that reduced form. The other thing that happened was that, and they say here that post-transcriptional effects. So basically, the aphid, because they have they have symbionts that help out with this, but they have enzymes and effectors and other immune system affecting proteins and compounds when they put their stylet into the plant tissue.
And just quick sidebar, oftentimes this actually is how aphids are sort of subspeciated, is by the enzymes they produce. It makes them a different color, or the enzymes they consume make them a different color, and so they're, or not, more so the species they are hosted on is different, so their enzymes are different, so they're a different species. But this is sort of how they can categorize different aphids, different production, different enzymes to oxidize and reduce.
Yeah, it's hard to really say. Like some species are going to act one way, some are others. Some are generalists, right? Some aphids are generalists, and some are not. Some are specialists. And what allows them functionally, ecologically, to be a generalist or a specialist is definitely related to symbionts that they have in their bodies. Like you said, enzymes that they have access to, endogenously, or that symbionts are doing, and that sort of a thing. And whether they can attenuate the plant immune response or not.
We looked at a study actually. The one, we can jump right ahead to it if you want. The one that what was it?
Is it redox control of aphid resistance through altered cell wall composition neutral?
Yes.
That was the one I already referenced. Yeah, that's the one we're
Yeah.
talking about right now.
Well, yeah, okay. But there was I think there was actually a, well, I guess I'll shut up. Go ahead. There was a part that I wanted to, I'm completely blanking. There's a part of it that I wanted to, that you brought up.
Make sure to find it if you have notes or something, because that.
Oh,
that
yeah,
sounds
no, my
important.
notes are a goddamn disaster.
Oh, no. Well, I only wanted to make one other point, which is that, they definitely emphasized in this research report that that there's a post-transcriptional effect. So the aphids are literally changing how the plant can express its genes, essentially, how the plant can upregulate or downregulate defense reactions, immune response, and things like this.
I remember what I wanted to say.
Okay, what was it?
The high BRIX thing. So you
Oh, sure.
You sort of skirted around this a second ago, but a lot of people will claim that, high BRIX equals pest resistance. And maybe that's true for certain pests, but I can tell you straight up from this research validates, and probably prior research, that, aphids are interested in these sugars. And now maybe as these sugars become in excess, it can be a resistance for the plant. But essentially, like, this is what the aphids seek out. High BRIX does not equal pest resistance in all cases. I just wanted to put that on the table. I think that's important.
It's important to me, and I think longtime viewers know that I kind of butt heads with this sort of concept that's kind of prevalent in regenerative agriculture, which is something I'm an advocate for, of regenerative agriculture myself. It's just like, I get it. A lot of people don't study insect physiology. It's not very common, not in vogue. It's becoming more and more easy to do because, we have the technology to be able to do things like genetic sequencing and things that we didn't really have access to in the past. But yeah, like I said earlier, like, these aphids want sugars. Now, it is true that plants, one of the main factors of photosynthesis is that they want to get more sugars assimilated, and they want to store those sugars and then use those sugars in other processes. So obviously, I think it's totally valid to say things like, higher assimilation of sugars is kind of showing you that the photosynthesis that the plant is wanting to do is happening. That's totally true. And you need resources in order to mount an effective immune response. But the other thing is that it's very well documented, and I'll cite some sources if people want. But basically, plant immune priming and response. Like a lot of people are ask me questions about, like, systemically acquired resistance, SAR, ISR, that kind of stuff. And the thing is that priming the plant could be really useful, and in a lot of cases is. But anytime a plant mounts an immune response or even gets primed to do so, it uses resources to do so. It's a zero-sum game to a degree. Those immune responses are going to cost resources that it would otherwise use to, like, grow larger and do other sorts of things. And sometimes those responses are things that make the end plant product that we want, like, better, like a malic, so acid sugar content in grapes is the common example people like to use. Obviously, cannabinoid content, terpene content, cannabis is another example of that. And so you could make the argument maybe that if you supercharge the immune response, or if you really emphasize that, if you can find some way to elicit it, you might get some real dank. But at the same time, you're necessarily sacrificing other physiological changes in the plant. So you just got to know what you're doing.
Yeah, and you this really touched on an important thing with growing. I've been indoors, I've been outdoors. As an outdoor guy, it's cheaper to do, but you are a victim to your environment, and so there's really no avoiding or getting around the fact that if it's 110 degrees out, and it is, if it's 10% humidity, and it is, those plants are not going to be as healthy as they could be.
Right.
They're having to spend a lot of energy to resist this heat. Produce. In a particular example, something we've done on the hill for a long time is last couple of weeks, you want to let that soil dry out and allow the plant to try and keep itself alive by producing more resin. And so that happens sometimes, but sometimes you just get this lack of production at the end of your cycle because of prior stresses that you were sort of unaware of. So my idea of a good grow is taking away all the stress and then, peppering the stress where it is advantageous.
No, Aaron, if your plants are stressed, you're just a bad grower, and your plants are bad. And they're not fit for human consumption, which is one of the most egregious claims I hear people, people parrot this quote. I just feel like you're gaslighting growers when you say that. And besides, if we're talking about holistic health, what is holistic health if you're not considering the environment in which the plant is growing? Like, what is that?
Good
How
point.
can you possibly talk about it? Your environment is a factor. You mentioned stress, oxidative stress, right? Oxidative stress from like heat in particular. I posted recently about elevated temperatures causing stress. Mutual friends of ours have even said that they grow their plants without any stress, but that's literally not possible.
Right.
Literally not
It's
possible.
literally impossible. I mean,
to some degree at least, right? And also in some cases peppers. In some cases, if you stress them out a little bit, like you were saying with the cannabis plants, you give them a little dryness in the soil,
Yep.
Increases the heat in the fruit. Well, maybe I want that,
Right.
but you're stressing out the plant.
Well, fuck the plant. Sorry, excuse my language.
Well, I'm gonna have to bleep that out for YouTube. I don't think they mind too much. They have they have lots of videos on there.
I like to keep it lively,
Yeah, right. That's what people want.
So we talked a little bit about antioxidants. What are some oxidizers? Now, I generally am familiar with the ones in the soil that oxidate soil: oxygen, nitrate, nitrite, manganese, iron sulfate, and carbon dioxide. But how does that work, and how can we sort of understand that in insects?
So things that actually oxidize, right? So
Yeah.
I have this reference here. It's called Detoxification Mechanisms in Insects, and there's a ton of references here that I'm happy to go over for people who have questions. And if you're watching this on YouTube in the future,
throw it in the comment. Yeah.
I'll put it in the comments or the description.
Yeah.
But here we have, so here I just highlighted these paragraphs here. I'm just gonna directly quote because I don't want people to I don't want to like say something that's not true, or I don't want to like misrepresent it with like a metaphor or something. So I'll be quick, but basically I think this sums it up really well. It says insects are faced with numerous toxins, which we call xenobiotics. So foreign biotic compounds in this case. As they go through life, some produced naturally by plants, sometimes we call these allelochemicals, and some produced by humans, like insecticides. And to survive the natural toxins, insects have evolved various detoxification mechanisms. These same mechanisms also sometimes allow insects to overcome insecticides, and the level and type of mechanisms differ greatly. This results in differing toxicity among different stages, populations, and species of insects. Knowledge of detoxification allows us to better incorporate chemical resistance mechanisms in crop plants, to better select insecticides that will be effective when applied. And they talk about two things. They talk about phase 1, which is primary, and phase 2, secondary processes. Phase 1 reactions consist of oxidation, hydrolysis, and reduction. So we already talked about some reducing and oxidation. So insects absolutely 100% have access to these tools just like plants do. I mean, it shouldn't be surprising that in like the 400 million year plus dance of evolution, this would be the case. Like, they learn a little bit from each other. Plants too, to some degree. That's why you have like effectors and effector trigger immunity, and then you have things that are deployed to affect that, and it's just this arms race. But that I didn't really answer that too much, but this paragraph here goes into more detail. So there's phase 1 metabolite. So mainly, to answer your question, this is done through like so there are metabolites, not just like chemicals that they're eating and things like that. Although when they are feeding on the plant itself, that you can have this like this ensemble of various chemical reactions that are happening. Cells are bursting, hypersensitive responses happening in the plant. Now I'm mentioning insects here, but this is true for pathogens as well, and I have a really great example of it in another article. Actually, I'm going to switch a little bit because it's on my mind, and I think it's really important.
Do it.
So we know that there are different kinds of pathogens, right? So here's an example. So, like ecologically, there are 3 main kinds. There's biotrophs that I'm going to talk about at least. Biotrophs, necrotrophs, and hemibiotrophs. So biotrophs are pathogens that have to interact or colonize the plant tissue, and they require a living host to work. They can't be on a dead host. They can't feed on dead tissue. They need to interact with living plant machinery to siphon out resources. Powdery mildews are an example of this. There are other pathogens like this too. And when plants interact with biotrophs, they're really effective with their hypersensitive response. That is when that's programmed cell death. So there's
is that apoptosis? Is that the same thing?
That is apoptosis. That is exactly right.
All right, learned something.
I had something on this here, right? I had this written down actually. But anyways, so what happens is that like the cells are burst. There's an influx, or actually it's called an efflux. There's an efflux of toxic compounds that come out of the cell wall, and then there's an influx of things from outside into the cell. So there's this like reverse pulling that happens essentially, that things come in, things go out, and that allows actually a cascade, an oxidative burst to occur. And that causes the cell to basically explode in then oxidative superoxides and peroxides and other sorts of oxidating nitrous oxide are released, generally speaking, and this denies the biotroph its host. Other things that happen that are triggered from these reactive oxygen species are like lignin and callose production. That causes the creation of like a protective barrier. So you have all these like dead cells that are like just oxidizing, and the biotroph is like hopefully has some sort of defense against this, and you kind of create a lesion this way. Mm-hmm. Now this is incredibly great against biotrophs that don't have adaptations to this. It's incredibly ill-effective against necrotrophs though. Necrotrophs and hemibiotrophs, they can switch between the two. They can just feed on the necrotized tissue. They have no problem with that. They don't need a living host. In fact,
is botrytis a necrotroph?
Botrytis is a necrotroph.
Yeah, that's one that feeds
actually,
on dead tissue.
Botrytis is a hemibiotrope.
Okay.
It's a hemi, so it can live. It can do both. It can. In fact, in some cases, botrytis can live in a plant. I think you've heard me say this multiple times on the Cheap Home Grow podcast, but it can live inside a plant in some cases without showing any visual symptoms, kind of as an endophyte. And then usually it's associated with like reproductive tissues. So as soon as that plant bolts, if it is an endophyte, then it will start to colonize, and you start to see like the mycosis of like floral petals or other sorts of
Right.
tissues like that.
Right.
Yeah. So a hormonal change, a chemical change, various changes in the plant somehow kind of like elicit this response, and the botrytis goes, it's time for me to do what I need to do. So,
Interesting. Yeah, I like the way you put that. Fido brings up a good point. Biotrophs tend to be extremely sophisticated and specialized due to this molecular arms race.
I agree with that completely. Yeah, and so like parasites are this way too, like aphids, right? Aphids, leafhoppers, things like this. It's not a perfect metaphor, but you can kind of draw a parallel between like aphids and sucking plant insects like that, and like caterpillars and things that are
generalized. Yeah.
That are just sometimes those are specialized too, but yeah, they're munching on the leaves. Grasshoppers, in fact, the
polyphagus, is that what it's called?
Yeah, so like a generalist, that means they eat many different things.
Okay.
But like I guess what I'm trying to say is that like a caterpillar or a beetle or a grasshopper has mandibles that crush and destroy and pulverize, and that's actually the ancestral insect mouth part, basically. Or actually, I would say I'd go further to say arthropod mouth part is sort of this mashing, gnashing kind of
physical crushing rather than a chemical crushing, or chemical
or rather the stylet, right? They're super adaptive.
I gotcha.
Well, plants weren't always vascular, right? We didn't always have vascular plants.
Okay.
And so there was no vascular system to exploit, and then there was, and then, the rest is history.
Yeah. Okay, I'm with you.
Yeah.
I wanted to talk a little bit about soil, if that's okay, just briefly.
Absolutely.
If I'm sure that you're aware, there's some examples of redox in soil. There's redox potential. Redox poisoning was an interesting subject I was reading about.
I'm not very versed in that. Do you have a?
My understanding was, so it's poised. This is my own. I rewrote this in my own so I could understand it. That's how I tend to write really complicated stuff.
That's fine.
Poised in the sense of having the oxidizer in both its oxidized and reduced state. So
like a superposition or something?
Yeah, exactly. Like you can think of it like potential energy.
Okay.
That's my
I don't know what you mean by that.
That's not from the paper. That's my brain saying this is kind of like potential energy, like potential versus kinetic. Is it happening?
Or yeah.
Or is it loaded?
Chemical energy can be like that, right? Yeah, you can have stored chemical energy, and so something has to trigger that effect. Well, actually, I don't know if this is the same thing, but you remind me of the fact that, like we were saying earlier in the beginning, sort of these reactions are happening kind of all the time. And so and even in some of the I think it was either in the Aphid research report I just talked about or another one that I will talk about later. Basically, they even mentioned they're very difficult to really assess the cell's like redox potential because it's constantly changing. Metabolic wastes are being excreted and processed, and other factors are coming in and coming out. And so I feel like we're talking about like poisoning and being kind of like both at the same time. I could imagine maybe a scenario where these reactions are kind of like happening either all the time or like at the same time, kind of, because these reactions almost like simultaneously, and it'd be very hard to track.
It's almost like trying to find the quantum position of an electron at any point in time. It's like we can kind of say that it's there in the general area, and that's actually quite relevant, right? Because that's what we're talking about, is electrons attached to atoms.
That's that is true. And then I guess you could start if I wanted this video to go viral, I would title it something like The Quantum Mechanics of Regenerative Agriculture or something. Right. Yeah. We all know, but nobody goes to a doctor and go, "Doctor, I have a problem," and the doctor goes, "Oh, it looks like some of your atoms are in superposition," or "Oh, it looks like your atomic structure is a bit randomized. Let me just get the de-randomizer out." It's a little bit No, it's a little bit much.
Oh boy, yeah, I like that though. That's fun. That's fun to think about. So there's redox potential, which is sort of hard to measure, but then there's the anoxic and aerobic, or first, sort of anoxic or aerobic conditions of the soil being measured in millivoltage, which can vary so substantially in within the soil, but there is a measurement to be made there. I think that, like, this is information we can gather and we can add it to the list. And what my research says is we add that to the list about right below pH. That pH is seems to be a little bit more dominant in this arena of, what does what. In other words, like, it's upstream. pH is upstream from EH, redox potential.
I can kind of get behind that, because it's kind of like we were saying, like, environment dictating the reaction. Like proteins in your body, like, you need a catalyst sometimes, and if you don't have it, then that reaction either doesn't happen or happens slower. So that kind of makes sense to me.
Right. Yeah, I like where this discussion is going. I think we should label it quantum physics of regenerative agriculture.
Yeah. And like Phytoalchemist is saying here, the rhizosphere alters those redox potentials, right? So, like,
right.
And I mean, like, there's, like, looking at, like, pockets of water in the soil, how porous it is.
Yeah.
If we had Brandon on here, he would be perhaps really excited to talk about those sort of esoteric intricacies of the soil dynamics, right?
Speaking of Brandon, you brought up a good question. I think his question was sort of relevant to the end of the or part of the nitrification process.
Oh yeah, he had mentioned about that, right? He thought that'd be a topic we could talk about. I didn't do a lot of looking into that personally, and it's definitely not my purview. But did you kind of understand? Because talking about nitrate,
yes.
Carbon ions. My understanding is that that's not as common, at least with carbon, like ionic. What's the word like for binding?
Like what do you have? Like, oh gosh, what is it called? Covalent bonds, like where redox is. It's the redox part of it is sort of like not very redox-esque, where there's not really an exchange of electrons, but they're sharing.
Yeah, well, and that like I guess you could say in an oversimplified way, right? It kind of it makes them unavailable, right? It kind of makes them more non-reactive that way until something comes along and either like shunts it or like binds it with more things, or those intricacies about chemistry. Once you start looking at, like, I mean. It only takes a couple of, like, I even have here some Wikipedia articles, which is of course not the acme of excellence here, but there is still a lot of references on this Wikipedia, like superoxides and the active oxygen species you can look into, and of course antioxidants and oxidative stress. So I have even these references here, and it gets super. I mean, you look at this free radical toxicity diagram on the oxidative stress wiki page. There's a lot going on, and even that's an oversimplification.
Yeah, and to try and give an example of, I think, what Brandon was talking about, something that's like, okay, so a simple example of how in the nitrification process, how carbon bonds to nitrogen would be urea. So urea is, if I'm not mistaken, carbon monoxide, NH2. So I'm not sure what that compound is, NH2, but that's urea. And so that happens during the nitrification process as part of this. This is, I think the specific question was, how does NO3 bond with things like carbon to make new stuff? And I think urea is one of the things.
Okay, yeah, okay. So, like, that is probably true. I actually couldn't corroborate that personally. I don't know all of those.
I know you'll do the diligent research when this is done, when you're editing it and all that.
Yeah,
so if I'm wrong, definitely throw a.
I'll put it up right here.
Right
if
I'm
able to find it. Yeah, right on the face. Yeah. But like,
So
but I mean, that's actually just a great reminder to everyone here that you can't know, or at least it's very difficult to know everything, right, there is to know, especially because we don't really know everything yet. And also, I want to say that I guess on that topic, different insects, different organisms, different plants, you know, their age, right? The nutrients that they're getting, their environment. These are all going to play massive factors like phytoalchemy. And that's not supposed to be some sort of like, you know, ultimate caveat so I can just explain away everything and be like,
no. But just to say
but
that
it's
there's so
true.
many things that you have to take into account to try and understand or get a holistic picture.
But I don't want to waffle too much.
Sure. One thing that's important to note about soils before we jump away is the a redox potential of, you can sort of find a dependable redox potential in aerated or aerobic soils of a positive 300 to 500 millivolts. And, you know, if you have the ability to measure redox potential, you can add that to your list of tools and do with it what you can.
Should we talk about how difficult that is?
Yes, please do, because I really don't
It's
know.
very difficult.
Please.
It's super difficult. It's super difficult. I sometimes disagree with some of the proponents, or rather, I think it's more so the people who are. I think the people who follow, like for example, Olivier Housson, you know, who has done a lot of research in this kind of realm about redox and that sort of a thing, especially particularly in soil. And you know, sometimes there's some like a crow or something that looks a little odd in the peripheral. But
He's
anyways,
watching you. Birds
but
aren't
yeah,
like.
it looks like it. Nature's on his side. No, but the thing is that
Olivier Housson is there's Olivier Housson is sort of like the I guess I sort of, when I listened to his stuff and I read his stuff, I found it to be sort of like the whimsical bleeding edge stuff. Like I enjoyed reading it. But
yeah, I have a
but
paper
I
right up
found
here
it
as a
hard.
reference.
I found some of the grounding hard to relate to. Not to say that like this is really good. It helped me understand it. I recommend people read that paper by Olivier Housson, the recent one by Olivier. But
Yeah, well, I don't know which recent one, but the one that I have here for people who are curious is called Redox Potential and pH as Drivers of Soil Plant Microorganism Systems, a Transdisciplinary Overview, pointing to integrative opportunities for agronomy.
the
And
only
but like,
problem I had with that, sorry to cut you off, just real quick,
sure.
was the drumming up of the focus of redox potential. And I get it, it's important, but it sort of like all the other research points at pH being a guiding factor upstream, and this was sort of opposite. And I don't have a problem with that. I think it's important to have this opposite perspective. That's how like good science is done. You know, you have people disagreeing. But yeah, I do see some holes I can poke in that, and that, you know, that's not to say it shouldn't be read. I think it was a great article.
Well, like, I was just gonna point out. Oh, there's another paper actually. There's two papers. One of them is just where he's just the author, and that was the one I mentioned. And there's another one where there's other contributing authors, and that one's called Leaf EH and pH, a novel indicator of plant stress, spatial, temporal, and genotypic variability in rice, Oryza sativa, and then of course Linnaeus, the moniker. And I think it's a really important thing to consider that particularly like in research in general, we can't extrapolate everything from one plant, from one research about one plant. It's just not good enough. And so I think that's kind of my point, is that I don't think the same thing sort of happens with me and Paul Stamets. Paul Stamets is a great guy. I like Paul Stamets' I like what he's done for mycology in general. I thought it was maybe, I don't know what, about naming a Star Trek character after him, and the mycology, the drive. Oh, spore drive. Spoiler alert! I'm not going to talk about that much.
Wait, what? They named
So
after him?
in Star Trek Discovery, this is a tangent. I'm going to be very quick.
You can edit it out for the live guys. Sorry.
The writers thought it would be good to have this thing called the spore drive, which is basically there's a character, Lieutenant Paul Stamets, named after Paul Stamets,
Yeah.
who is basically he is an astromycologist, and apparently, I guess Star Trek made panspermia canon. I guess there was an earlier TNG episode, and the real OGs know what I'm talking about. That might have actually kind of hinted at this in the universe, but I just think it's sort of like, come on, man. There's like a dark matter mycology network, and so the spore drive allows the ship to like dimension hop through.
Okay, so they're trying to connect
mycelia.
They're
And
trying
I just
to
think
connect mycelia to dark matter. Basically, they're making this.
It's kind of like that. There's a character who like dies and then is colonized by it, and then comes back to. It's just. I don't know.
I can't believe I didn't see that. I feel like I should have watched that in preparation for this.
Dilithium crystals and crystal energy coming from the engines of the Enterprise and that kind of stuff. That's also not really doesn't make sense either. But at least it kind of, I don't know, I can kind of wrap my head around that as like a fuel source better than that. But anyways, my point I'm trying to make earlier to go off get back on the tangent, that I think people read these research reports by people who are prominent authors, Busan, Stamets, whatever. And then I think people make extrapolations, just like I am actually, and just like you are here. And I think sometimes they are a little bit narrow, and I think that people just get excited, right? And it's just not necessarily the case that you should base everything on it. And like we were saying earlier, one of the reasons why Redox is kind of maybe not the best tool, or maybe not the most simple to utilize, is because it's very difficult to test. And you reminded me that I wanted to talk about this particular passage, which I've highlighted here in the report I just mentioned. It says, so however, I guess I should read the first sentence before. So the current increased understanding of reactive oxygen species acting as signaling molecules has opened new avenues to exploit redox biology for crop improvements required for sustainable food security. However, these works on redox regulations remain largely at cellular and organelle levels, despite some advances and a growing interest in the understanding of redox mechanisms of plant systemic responses to pathogens and abiotic stresses. An understanding of redox regulation at both plant level and field level is still needed. And in the materials and methods, they go over how they go about measuring redox, and it requires a really complex what's the word for, like a meter, essentially, that uses like platinum plates, and you insert it, and you're testing. First of all, it's an abstraction. There's no way, it's not possible really to track the absolute redox processes. It's kind of like we said, it's always constantly changing. It's very dynamic, and especially it's dynamic in living organisms rather than like, not to say the soil isn't alive.
Right,
but
that interaction happens inside the microorganisms within the soil more than anything than it does in this it's an interconnected system that we're examining. But as we dive deeper, the insect is the place where we find really prime examples of redox in terms of how it's learned to function to sustain its food source or adapt to its food source. Oh, that was a cool thing. The plant acclimatization through that was the study you posted.
About the temperature change, right?
Fight hard or die trying when plants face pathogens under heat stress. That was really cool.
That's a great title too. It definitely sticks in your head with that title, but it's true, right? And like, for example, we talked about biotrophic pathogens and necrotrophic pathogens, and some biotrophs might be negatively affected by increasing temperature in the environment. Some of them might be more adapted, in fact, to higher temperatures. Now remember, in Earth's history, sometimes the temperature has been higher and sometimes it's been lower in various places. right? And so over millions of years, you adapt or you die. Necrotrophs, on the other hand, might be because they don't worry about the biology, right? It doesn't matter to them so much to interact with the physiology of the plant. They might not worry about it too much. They can maybe be more active at higher temperatures, and if the plant's already stressed out because of high UV radiation and temperature and certain physiological processes break down over time, then it's going to be real bad for the plant host, but it's going to be no problem, no factor for the pathogen. And as this continues, the pathogens that get really good at this are going to do way better, and the plants that get way better at this are going to do better, and the ones that don't, well, they will fail.
Those are the plants in your backyard that get bud rot, or not your backyard, but in general, those are the ones that get the bud rot, that get the PM, that that get the problems that aren't necessarily designed for that type of resistance for your environment. Maybe they don't have those oxidizing enzymes to protect them from aphids.
I mean, on that note, just as a reminder, like the plants that we consume are not natural. They're not native. Like we have done a lot to plants, whether you want to talk about genetic modification, whether that's in a lab or through artificial selection. One reason why insects are super attractive is for the same reason we are. Sometimes you get natural hybrids, that's true, but by and large, you look at what a banana used to look like before humans got at it, or what an avocado used to look like before humans got at it, or various berries.
Cauliflower didn't even exist, I believe, before humans. I think we
right.
Yeah.
All of those brassicae, right? All those brassicas, right, like Brussels sprouts and all of that. So yeah, so it's almost like you can't even talk about. It's not even like a natural interaction. An untouched by human interaction is my definition of natural here, right? It's not like there's wild type plants with all of their additional defense compounds that we've read out of them unintentionally, or in some cases intentionally because they might have been poisonous. Hello, tomato. People who know me know that I can sometimes be sensitive to them, although I do like their flavor. Unfortunately, the solanine is a little bit problematic for me. And other people have various reactions and allergies to different plants and different things in them. And so,
and that just goes to show, the biology, the evolution of humans is parallel that of insects, man. We're all doing the same thing at the same time, trying to adapt. All right. So speaking of that, I have a question that says, some pests can release oxidizing enzymes to control plant growth or increase toxic substance production in the host. So that's adding a level of diversity there that you sort of touched on earlier when insects create a gall. Yeah. So if you could maybe touch on that a little bit, maybe some examples of ways that pests can increase toxic substances within the host.
So, the physical destruction of tissue creates an immune response, right, in plants typically. And those responses are going to, there's local responses that happen more quickly, and then there's more systemic responses that radiate out from that occur over minutes, over hours. But it's important to note that at least for some pests, and it's going to be cultivar dependent, it's going to be individual dependent on the insect, and different populations are going to be better or worse in aggregate. But basically, like I said before, those insects and pathogens will be able to attenuate this response too. And in the Future Cannabis Project, we did there's a presentation about IPM recently with 4 other people, and I wanted to be quick about it, but I did kind of want to bring this point, and I had this great graphic I don't have right now that talks about how these responses occur and how quickly they occur, but also how slowly they occur between when you elicit it with just the compound and when you have the actual pathogen or pest actually eating the organism, because they can release those effectors and attenuating enzymes and other sorts of things that make the immune system less functional, that like disrupt it at various points along the path. But like essentially, like sometimes insects rely on that reaction in order, or not necessarily a reaction, but just the built-up toxins in the plant, and they can sequester them. So I feel like anyone who's going to make the point that like plants can be like whatever completely resistant means, you know, like what is the mechanism of that?
Right.
I actually have a this might be, you know, this is actually a video. This is a John Kempf video actually. And if I play this small section for you, it says level 4 of plant health. And I've always been interested in what was meant here. It says plants develop increased resistance to the entire beetle family. Well, it's an order, but that's a minor point here. So they include several beetles here: Japanese beetles, corn rootworm beetles, squash bugs, which are not beetles, Colorado potato beetles, cucumber beetles, and the marmorated stink bug, which is not a beetle. Then nematodes, like root rot nematodes and viruses, which are not beetles. But I guess he's talking about multiple things here. But he says this.
At this stage, plants become resistant to all of the beetles: Japanese beetle, Colorado potato beetle, marmorated stink bug, and so forth, as well as nematodes and viruses. So nematodes do not have a chance for plants at this level of plant health, and viruses can be present in the parent plant material, such as in seed potatoes, but when the crop is grown out, that viral infection will not express itself, and in fact the viral infection can be removed entirely when we have the right mineral nutrition to accomplish that.
Now, I've never heard that in my life. I've never read anything that sort of supports that, and there's no reference that I can go and check this information from. And you know, I'm not trying to like pile on to somebody who sometimes I butt heads with about claims, but I do think it's really important to consider. Like again, some in this video, for people who want to reference it, it's called Managing Crop Nutrition versus Pest Management and Sap Analysis, John Kempf, Regen Rev 2021. So it's a recent video. And there's another part in here, and I'm not going to go search for it, but basically he's talking about how plants have resistances because they have waxes and you know, physical basically structures that prevent the like a pathogen or a pest from like affecting the plant. And it's true that there are physical structures that have a resistance component to them, like the bark of a tree. Some insects just can't get through it, so there's no, that's called non-host resistance, for example. They're just not a host. They're not suitable food because there's no way for the insect to exploit that or whatever. But you know, it's very trivial to look up some of the proteins and enzymes that fungi and bacteria and even insects can utilize to break down plant defenses, like cutin. Cutin is the wax that's on the leaves. In some plants you can see more, sometimes you see less, but most plants have some sort of a cutin layer. Well, cutinase is an enzyme that breaks down cutin.
Is that how you say it? I've always said chitinase.
Chitinase is chitin and chitinase are totally different.
Oh, different things? Oh, good. Okay. I've never heard this.
Cutin is a wax.
Okay.
And so like you know, that has a lot of, what?
Does cuticle, like the root of cuticle, have that? Okay.
Yeah. It's kind of like, yeah, it kind of works like that. Like it's the cuticle of,
sure.
So that wax has numerous effects, right? One of the effects is that it helps keep water inside. Water retention. Some plants really rely on this more than others, depending on the environment they live in. And of course, it does have an effect of washing away water, and potentially even like helping that water kind of drip down and not stay on the leaf where a lesion or a pathogen could get through. Remember that typically on the phylosphere, you've got microbes that are on the leaves just kind of hanging around, doing various things, making biofilms, that sort of a thing. Sometimes the stomata opens, water comes out through evapotranspiration, and a bacterium goes into that xylem. Maybe it's a good guy, maybe it's a bad guy, but that wall literally opens up multiple times a day. And so, like, how can you not have things enter things that, and some microbes like Pseudomonas syringae produce, I think it's carnitine or quarantine. I'm not saying it right, but it's a phytohormone, and it tells the stomata to open. So some bacteria and other organisms like pathogens, but also beneficials, use the same pathways to enter into the plant. And I guess maybe I went on a tangent. That wasn't quite what you were asking, was it?
Well,
Do you even remember?
no, I do remember about the toxic substances. It's not that not that you didn't answer it, but you definitely had a nice discussion.
Yeah, I guess so. I guess so.
It's relevant. I don't know about directly answer. I think I asked for exact examples, but that's sort of, maybe I got you on the cuff a little bit.
No, you didn't. Monarch butterflies are a great example. So,
Okay.
like, caterpillar. So the caterpillar munches on the leaves. It already has certain alkaloids and things in the tissue. It sequesters those poisons. They're supposed to be poisonous to it, but it's adapted to not actually be affected or not be affected very much. And then it literally takes those poisons and it puts it in its own tissues, and it stays in the tissues even when it's a butterfly. And so the
Cool.
butterfly goes around, and it has aposematism. So it's got bright colors that tell other things that it at its peril will go and try to eat it. And then over time, things learn to not do that.
So it actually contains this volatile, like, poison in its cell structure that doesn't harm it by consuming it. Is that correct?
That's right. That's right. And a lot of insects do something like this, where they sequester it. The spotted lanternfly is a good example. It's a huge pest in the East Coast, and I think it's only a matter of time before it becomes a pest in the West Coast. Unfortunately, it's just kind of the nature of a lot of pest abatement. Not to discount the USDA or any other, or CDFA or anything like that, but it's a very, very pernicious pest. And when it feeds on it's a preferred host of Tree of Heaven, and I have videos on the YouTube channel about that. It'll be even more successful because it can sequester these very toxic alkaloids. And some people have asked me, well, aren't there natural organisms that take control of it? And there are parasitoids that will parasitize the nymphs or the eggs, for example. And
nature always has an answer.
there is an answer there, right? Yeah, but a lot of things don't like to eat the adults because of those poisons.
Sure. Yeah, so it definitely puts their survival rate way up. That's very cool.
And,
yeah, good example.
Some plants will produce more in response, but it doesn't matter because the insect,
right,
already has a defense against it. So
hide that back. Okay, yeah, that makes good sense. Yeah. Jumping into the paper from Krishnan and Kodrik from 2005 about the Egyptian army worm.
Mm-hmm.
It was cool. I believe that, so if I read it correctly, the armyworm adapts to its food source using this redox, and this is sort of like this would be like an empirical site for.
Oh, is this Egyptian?
Yep.
Armyworm?
Yep. It was the Krishnan and Kodrik.
I have the page up here. Spodoptera litoralis. Antioxidant enzymes in Spodoptera litoralis, are they enhanced to protect gut tissues during oxidative stress? And what's the answer?
Yes.
Yes.
And that's very interesting, right? So these armyworm, this is just an example, I suppose, and an actual paper that has found that this is how this redox and adaptation function in nature. Kind of cool. So I don't know if there's so much of a question that I can form out of that, so much as a reference.
Well, I have this. There's this line here in the abstract that I think is relevant to what we were talking about earlier. I don't know if I went into too much detail, but basically, different sections of the insect gut can have different pH levels, can have different enzymes being produced or being concentrated a big part of insect growth in general, and particularly things like caterpillars and larval like stages of insects, is that they got to bulk up really fast. They got to eat a lot, and the more they eat, and the more they can process what they want and get rid of what they don't want because it's poisonous, because it's metabolic waste, of course, in various other ways. Like for example, going from aphids or going from caterpillars to aphids real quick. Aphids, they feed so much phloem, they can't even process all of it. They're like a little pump, and you've heard me talk about the tap analogy. They're like a little tap in like a maple tree, and they're using the pressure, the turgor pressure of the phloem channels to just shunt that through them, and the honeydew gets produced. Even the honeydew, believe it or not, has microbes in it, has effectors in it that affect the immune system, and that sort of a thing too. But here we have, it says here, antioxidant defense was measured by the activities of superoxide dimutase, catalase, ascorbate peroxidase, and glutathione sulfur transferase, peroxidase, and a significant increase in the superoxide radical production in foregut tissue, foregut and midgut contents, concomitant with an increase in total peroxide and foregut contents and protein carbonyl levels in foregut and midgut tissue, which I know is technical, I'll talk about it, were noted in larvae fed on the plants. In contrast, those fed the semi-artificial diet. So they had them fed on an artificial diet, and they had them fed on the plant.
It was a potato plant, right? Is that right?
That's right. That's right. Well, actually, it was the potato plant. But they talked about the Solanaceae and the Cruciferaceae, so cruciferous crops and solanaceous crops. Where was it?
So you were at the semi-artificial versus the artificial, the potato plant.
Similarly, a significant upregulation of antioxidant enzymes like superoxide dimutase in the midgut tissues, catalase in the foregut midgut tissue contents, and ascorbate peroxidase in foregut contents, midgut tissue and contents, and also the glutathione sulfur transferase peroxidase in foregut tissues was recorded on the plant diet in comparison to the semi-artificial diet. And if I remember correctly, when I looked at this report, one of the other things they talked about and mentioned was that there's no way that just these enzymes contribute to these effects. Those effects, namely being the production of these antioxidants. So other factors are at play as well that they didn't really test for. And this is not surprising. Things like microbial symbionts could be a factor. Things like actually the chemical constituents of the tissue of the plants that they're feeding on themselves. We talked about earlier how plants can affect the nutrient absorption due to redox reaction and reduction and things like that.
And it actually goes on right there to say the pro-oxidant allelochemicals in the plant diet are thus eliminated by the insect at the expense of upregulation of antioxidant enzymes in response to increased oxidative stress from oxidizable allelochemicals. So there you have, you break that down, you're going to do a much better job.
Basically, so the allelochemicals, again, are the bad stuff. They're the poisons and toxins that the plant produces. And so essentially what happens is that these defense compounds can be oxidized and destroyed, essentially, or neutralized in a way that makes the plant tissues palatable and edible for the insect. And interestingly, when they eat an artificial diet and they don't eat the actual plant tissues, this effect changes. Although to some degree, even on the artificial diet, they do produce some of these things. So there's endogenous production of these antioxidants. And I think that's kind of important to mention, that there's sort of at least two things mainly happening. There's endogenous production of antioxidants that are meant to destroy toxins and sort of nullify the defenses of the plants. And then there's also effects that are happening, maybe that are not endogenous, that are happening from the external environment.
Consuming the actual plant material itself.
Exactly.
Yeah.
Exactly.
Yep.
So again, like I think some people misquote the research of people like Olivier Housson and other folks that make sort of claims, or maybe not even misquote. Some people just make erroneous claims about insects not having the enzymes to break things down. And
Right.
with that, I'd like to have this quote here, because I actually recently came across a couple of people who thought that maybe I was banging on about something that people weren't actually saying about perfectly healthy plants or whatever. So to immortalize it, I have this quote. This is also a John Kemp quote. See if you can see the part that I disagree with.
Our systems, our digestive systems, do not have a cellulase enzyme. And in an analogy that is similar to that between a dairy cow and our digestive systems, what is food for us is not food for aphids or for flea beetles or leafhoppers, because they do not have the digestive enzymes that are required to break down that last peptide to peptide bond that gets used to build complete proteins. They are completely dependent on specific forms of sugars and specific forms of soluble nitrogen, whether those be specific amino acids or soluble forms of nitrogen such as nitrate and so forth, as a protein source and as an energy source. So it's really quite simple. If plants are being consumed by insects, then they are not fit for us to eat, because they are so unhealthy that natural ecosystems want to take them out of the system before we can eat them.
I don't even know what that means.
Okay. I was gonna say, oh, there's nothing wrong with that until the very last part, and then he lost me.
Right, like definitely insects eat things that we eat. Like how can they not?
Right.
And also just for people who are maybe agree with this statement and aren't aware, but like we are animals, Aaron. Insects
You
are
are
animals.
too? Oh, I thought it was the
And
only one.
believe it or not, we actually share some,
Yeah.
not all, but some of the
Enzymatic.
same enzymes
Yeah.
that help us. We share that ancestry long time ago. The split was very long time ago, but we do share some of those enzymes. Even today, you can see the same genes that are in us, other animals, and in insects. So it's totally incorrect to say something like that. I don't even know what
Thank you
this means.
for touching on that. I think that's interesting and important. So very cool.
Yeah.
Sidebar: watched some cool program the other day where this woman in this native community chews corn and then spits it out. So she's producing this amylase, I believe, in her mouth. Like that you can produce this enzyme in your mouth when you're chewing on it, and then you spit it back out, and she makes that into like an alcoholic beverage.
I've heard of that. Yeah,
So
I have heard of that. Yeah, kind of baby bird it.
Yep. It was very natural, so it was cool. We can move on. That was just a cool thing that I learned the other day.
No, actually, you know what? I love that you brought that up. It's actually a great segue into this, or from this part actually. Most people are aware of this, but our saliva has proteins and enzymes that break down food. So like the digestive process occurs, it starts in the mouth. Most people have heard this, but just to reiterate, it starts in the mouth. We masticate it, we chew it like a grasshopper would, gnash at the tissues and really break them down mechanically. But we also have chemical digestion that's already happening before it even reaches your stomach.
Right, in the esophagus, I mean, all the way down, there's going to be different enzymatic production, right?
Yeah, and in fact, I'm a big nerd about various aspects of biology, and one thing I'm a huge fan of is venomics, the study of how venom, kind of is sort of like, how does it occur? How did we go from not venom to venom? Where did that come from?
Yeah.
And believe it or not, at least in a lot of animals, like spiders, arthropods like spiders and insects, but also in like reptiles and things like this, it tends to be the case that it is from saliva. It's salival proteins and compounds that over time they start to change, they start to get more complex, and they become really useful. So originally they were used for digestion, and we know that with spiders, like, when they bare their fangs into a host, they're externally digesting it, but also those toxins are also killing it, of course. So you're kind of doing both at the same time, and then they can just drink that back up. And a similar kind of thing is happening with reptiles and venom. And so I guess I just feel like that's kind of a cool example of how saliva and things that have one effect can get co-opted for like defense, just like how plant defenses can get certain aspects of plant physiology can be co-opted for defense, or certain aspects of insect physiology that might be nominally for digestion and absorption of nutrients become kind of an offensive weapon as well.
Sure.
Mm-hmm.
Yeah, and in fact, nothing really gets by the hammer of evolution without having multifaceted purpose, right? Like you better be a Swiss Army blade as a trait to survive.
Yeah, I mean, as long as you live in an environment where that matters. I'll give you one cool example of that, which is that if we go back to our aphids, tons of aphids and other insects have symbionts, and also plants for that matter, and fungi. But one of the cool things about that is that certain symbionts, specifically like bacteria, in like aphids and stuff, they have a very reduced genome. And in fact, in some cases, we can even tell what the ancestral, I guess you could say, free-living microbe was before it, where it evolved and co-evolved with like aphids and whiteflies and stuff for like millions of years. And what we find is that when we take a look at what that ancestor is and what the symbiont is, the genomes are like this. And it's because the facultative symbiont, maybe over time, the free-living microbe is taken up, and every time the insect, like simplifying it, but every time the insect takes it up to the environment, it benefits it. And so there's a co-evolution effect that occurs there. And so over time, the symbionts that go from free living to facultative to obligate, they're obligate symbionts. They can only live in the pest. They don't need all those extra things. And so evolution, evolution is a lot of things, and one of the things isn't as simple. But to oversimplify it again, a lot of times, if you don't need it, if you don't use it, you lose it, especially if it's physiologically costly, like having flagella that allow you to move about the environment. That's a ton of resources that you don't need if you're going to be a symbiont. So when you lose them and it doesn't affect your fitness, then it doesn't matter. And this is also how endosymbiosis happened to some degree in our own mitochondria, or in the chloroplast in plants used to be cyanobacteria.
Mm-hmm.
So really, a very long time ago, cyanobacteria got put into plant cells, and those are the things that are doing the photosynthesis that helps the plant host that we eat. So really, we owe everything to those really hard workers.
When you start tracing it back that far, it's like, it makes you wonder about that first redox reaction. What was the first thing that happened on Earth that created life? And you have to think about you have to think about it like a battery, and the potential exists once you apply electrons. Once you have, every photon has, I think it's like 10 to the negative 36 electrical charge per photon. Okay, so throw that into the equation, and you have some amino acids, and you have some proteins that are sort of the precursors for life. And you have to imagine that first redox reaction, that first electron transfer that created such long chain. Man, I might be misdescribing this, but
Well, there's some theories about that. There are some theories about that. I don't want to cut you off though.
No, please do.
So basically, you know me, I love all this kind of stuff. I've looked into things like virus origins. Where did viruses come from, and what do they have to do with us? And a lot of the research into that have kind of brought us some theories. None of them, or at least for most of them, they don't. And I have videos on my YouTube channel that go over some of this. Like my, if you're interested in this, you can check out my Global Integrated Pest Management Review 2019 parts 1 and 2, specifically 2, that goes over virus origins. Because I go over all of the various microbes that interact with cannabis. But basically, there's this thing called the RNA world, which is this theory about how the world lived before cells. And things like viroids that don't have a capsid, a protein shell around their parts, kind of make us think, well, hold on, maybe there was life. There is RNA, because some RNA can exist as can act as like a enzyme, essentially. I think that's the right way to say it. So not only can it like encode for the proteins and other sorts of things, it can also encode and have like a catalytic effect. And so it can like do things specifically, and also it can encode for other stuff.
Change things. It can move move electrons, essentially.
Well, it can just be like its own kind of enzyme to do stuff with. That's a very it's a very ham-fisted way to explain it.
Okay, I get it. Self-reliant.
Oh, kind of. But basically, this RNA world may have existed and may have predated cells. But one of the things is that at some point cells had to be produced, and we think that maybe there were like lipid encapsulations of things like RNA and things like various like little like you say like molecules, like proto molecules, I guess you could say. Well, not really proto. That's from a TV show, but these molecules over time, they got encapsulated, and there's various ideas about how hydrothermal vents are one possibility, because there's enough heat there that certain reactions could happen for the first time, speaking of like redox reactions. And perhaps viruses were some of these RNA that basically they were able to enter into these lipid capsules, protozoa. Yeah, and basically make use of them, colonize them, and then kind
create the cell wall, basically.
Well, actually, just destroy it, and then go on to the next one. So like, kind of like the first parasite.
Okay, that makes sense functionally. Okay, I get it. Okay. Oh, man.
But there's other theories too, and I think it's really interesting. But I love how you put it. Like, obviously, like, some things have to happen. I was going through some storage recently, and I came across a book when I was a kid. I remembered very explicitly the title. It's called "How Did Life Get There?" And
cool.
And this was like wrote in the 1980s, and it was talking about, and I think it did mention things like volcanoes and hydrothermal vents, and even nowadays we've refined some of those thoughts and cast away some of the things that don't make sense. But I do feel like the intense heat, and like, the cradle of life being water and the ocean, I think those must have had fundamental effects. So redox has always been there. Yep,
it's the first.
It's the physics. Life has to have had, there had to be a physics, it has to be what do I want to say? It has to be congruent with how physics works, right? Like the same physics that affect us now.
Right. So, electrochemical physics of the brain are highly complex and very sort of, to me, I studied psychology for a while, and a lot of this stuff sounds a lot like that. It's like, well, we don't really know, but there's this interaction between, very, very minute electrical charges and chemical release and uptake, molecular.
Mm-hmm.
So this is, as above, so below, fractally everything.
I love that you put it that way. I've been on a big Daniel Dennett kick. He's an American philosopher. He's still alive. And he's talking about a lot of things. One of the questions he asks, which I think, because he does a lot of theory of the mind philosophy, and he also uses like what we know about scientific understanding of physiology and physics to kind of you know, it's an important part of the philosophical thought process, at least for a lot of philosophers nowadays. And so when he asked this question that I think is really great. And since you kind of touched on this, I think actually, genius that you are, and that is, how do we get a top-down mind, kind of like ours, from neurons, this wet computer in our brain? I'm paraphrasing him a bit, but like people like to talk, even I have myself made comparisons between computers and our brains, and there's some similarities, but
They're opposite.
well, but crucially, computers require very strict, binary ones and zeros, very strict parameters in order for them to function. If some of those parameters don't work, it's all gone. It doesn't work.
Mm-hmm.
Neurons, every individual neuron, it's its own little thing, and it's making its own electrical charges, and it has its own fitness. It has to be.
Yep.
So how do you get from biology with cells? How do you get both superorganisms like termite mounds and termites that work in sort of like a collective, and you get a mind like mine or yours or some architect? And he brings up, he loves to bring up this this diagram of like I think it's a French or Italian architect, and like they did like a church, like I can't remember if it's a Bastille or something, and then like a termite mound, and it looks very similar. And of course, that's a little bit arbitrary, but he's casting a great question into the arena, which is like, how do you get both?
Right.
How do you get both from one?
I think we're discovering it. I would say that it happens at the very small. Happens at the very, 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 very small. So quantum computing, right, which is the ability of electrons to be in superpositions, and the computer can take advantage of that. And I think we're going to find that this super small, the scale of super small, is going to be the root of consciousness. Okay, I'm going to get into too much here, but it's going to be the root of consciousness. It's going to be the root of this sort of, I don't know what I'm trying to get at here, but
I think that I get the sentiment. I don't want to put your words in your mouth for you, but it kind of reminds me of that picture of people, like, it's super popular, and maybe it's a little bit of an old meme right now. But there's like a picture that traveled the internet of like a galaxy or a nebula or something, and then like a neuron, and like they like, sort of, superficially they look very similar when you overlap them, and I think that's a little bit of a stretch, but metaphorically, I mean, because that doesn't mean anything on its surface.
Right, but it does.
It's a great metaphor, and I think there is a lot of truth. Phytoalchemists even said the very big in response to you saying the very small.
Yeah.
And of course, physics has to do both. Physics must account for both. And so similarly, like you're saying, the minds like ours and the minds like termites or ants or other
from the very, very small.
Yeah, well, they have to be what's the word I'm looking for? Yeah, well, I guess I keep saying it. Yeah, they have to account for both kinds of physics. So
yeah.
both physics effects have to be in play. And
quantum physics and relativistic physics, I think Phyto is, I think that's where we'll find the answer to this, discussion of how we can move things forward with computer development.
Very possibly. He has some points about AI too, but I think we have gone on a bit of a tangent.
Yeah, let's jump back. Let's jump back into
in the comments a bit. But it's a great little interlude. I'm gonna put this part of the YouTube video and just be like, this is the philosophy section. Are there other parts that we want to kind of touch on? I wonder if the comments have some questions here.
This was really cool. I didn't expect us to get here. The answer is 42, he says. Yeah, I agree. The answer is 42. It's like my favorite number. So, do you want to sort of sum things up here? Do you want to kind of take see if we can pull some takeaways from this, and because we're about 15 minutes left here before I got to sign off and go water these plants in this 100-degree weather, 110-degree weather today. But what can we take away from this in terms of, the things we've discussed? Anybody fast forwarding to the end to get the conclusions, right? What do we tell them? Yeah, what do we tell those people?
Well, I think that the main takeaway that I would like people to have is that, maybe not surprisingly, redox reactions are fundamental. They're a fundamental aspect of life. They're a fundamental aspect of, like, even not like abiotic interactions are redox too.
Like
so,
fire or taking a dab. I'm going to redox a dab real quick.
You're gonna carboxylize the THC, right? I mean, it's like like I said, it's the fundamental facet. One of the fundamental paradoxes of life is that we rely so much on oxygen. So much of life does. Some things do not, but so much does. And somehow we've been able to keep those sorts of things in sort of a balance. And again, if I wanted to use like esoteric terminology or something, I'd say it was like yin yang, and it's like what they said in the Bible or something like this. But
I'm okay with all of that, I think, because it is sort of a holistic approach, and it's all we have. We're human, right?
What do you mean?
Well, we see things through the schemata of our perspective.
Yeah, that's true. Yeah. And if metaphors help us, nobody's out there, like kind of with the doctor example, nobody's out there like a robot. Although this might become racist in the future, but nobody's come so far. Judge me for my time history. Judge me for where I was in time.
Chronologically,
chronologically, right? Yeah. But I do think that there are processes that are happening all around us that are sort of fundamental, and they affect abiotic interactions and biotic interactions, and even that is an arbitrary. At the end of the day, even that is kind of an arbitrary interaction. I think getting to the heart of the fact that in a holistic perspective, it's all interconnected. Whether you want to attach some level of supernaturalism, I'd avoid it if possible, unless you have some evidence for it, in which case it wouldn't be natural. But anyways, or whatever.
We call these extrapolations, right?
Exactly.
Go ahead.
Okay, so I should actually summarize it. This will be the actual summary part.
Sure. Okay.
Basically, I want people to take away the fact that redox is hard to measure for cultivators currently.
Mm-hmm.
A lot of the redox research that's coming out now is kind of in its infancy, and I think is also somewhat plagued by, I think it would be maybe a little bit over egging the pudding to call it pseudoscience, but
romanticism.
Romanticism is a great way to put it.
Sure.
And I think it's also, I'm not an expert in it, certainly, so in some ways I'm unqualified to even talk about it, but I do know it affects integrated pest management, which is my specialty. It affects plant health, of course. It affects animal health. It affects the pests. It affects the beneficials. So in that way, it's affecting everything here, and particularly with microbes in the soil, inside of the plant, on the plant, around the plant. These interactions are happening, and if there's a way that we can take advantage of this, then we should do it. Right? We should try to do that, but we should recognize that at this moment in time, it's kind of ephemeral. I liked how you put it. It's sort of ephemeral and a little bit whimsical.
Right.
And it's kind of something I'd like to see a lot more research in, and I'd like to see a lot more practical application.
Absolutely.
Along with things like nanobubbles, that's a cool concept we could talk about in the future, or other sorts of things like this. Cool. I think there's some really cool vibrations. That's a loaded word, but acoustic vibrations, sonic vibrations, Mechanical vibrations, I think, would be an interesting topic as well. Very much romanticized in a lot of ways, but also I think have some real practical applications. Light, of course, we've talked about different spectra, UV light, infrared. I keep posting about helmet mounted thermals and like taking out pigs with like infrared vision. Like there are great cool applications of the physical world, and we're just being able to like use them for our human salt of the earth purposes.
Right. And as they work their way down the chain to guys like me, us lowly farmers, we'll start to have these tools available to us. Maybe my kids or my kids' kids will be able to measure the redox potential in their soil daily or hourly or something.
Mm-hmm.
But here's where we start. Here's where we start to learn about this stuff to be able to use it in the future, right? And that's sort of what this was all about, to say this is where the state of redox as we know it as farmers or as insect specialists is. Right?
Yeah, and if people have more, and people who watch this video who might agree or disagree with certain parts that we talk about, I would love to get new evidence, new research, new empirical studies about this kind of a thing. Please feel free to send them. If you're watching this on YouTube, put in the comments, some sort of a reference.
Yeah. Let's start a discussion on this stuff, right? Like, yeah, totally. Yeah.
Don't roll over for no data. I say it a lot, and I'll say it here.
I like that.
Just because somebody says it very confidently, or because somebody is very, I guess it's the same thing, being assured of themselves, or particularly if they have a service or if they have a product that they guarantee will increase your plant health, ask them how. What's the mechanism? How does it work? And if they can answer that question in detail, and specifically if they can cite sources that are separate from them, outside of them, then that's awesome. And please share it.
Yeah, absolutely. Yeah, and I appreciate everybody who jumped in the comments and added, especially Fido, Soulshine. You guys all were great contributors, and I know I'm missing some people in here that helped out and added to the conversation. So Devin Rash, this was a lot of fun, and to be honest, I was awfully intimidated by the subject.
Me too.
Yeah, good. Well, I'm glad that we tackled it together, because I was one of the guys who, when this shit came up in chemistry, I was like, what? I'll learn this the day I have to, right? So here we are learning it, and so it was a lot of fun, man. It was a shot in the dark, but I think we tacked down a lot of stuff. We nailed some things down that people can take home and chew on and develop new research from. Right?
Absolutely.
Yeah.
I honestly wish that we could take these comments in the IGTV put on YouTube, but
sucks that they don't show up. Maybe what we'll start doing next time we do this is we will repeat the question before we answer it, and that way it's kind of it's there, it's verbalized.
Yeah, I like that. It'd also be cool to bring on multiple people on the live stream. I don't know if we can do that either. But
I know I've been trying to get to my demise, I will get Fido to do an interview with me, but the guy's so gosh darn busy that I can never actually get him a free moment. But wealth of information. I would basically be silent if Fido were here. I think I'd just let you guys go. So, but yeah, I would love to have that happen. We could expand this conversation a little bit to, people that three, but yeah, to people that maybe know a little bit more than me. I'm not going to say more than you, because not many people do. But that would be fun next time if we could do that.
Yeah, let's do it. Well, in that way, I don't want us to be like cut out for no reason. So I'll end it here. Hopefully, this will be on IGTV and then also YouTube. If not, then I hope you got your takeaway, because that's it. That's all there is to it. We'll just do it another time, I guess.
You usually get this stuff figured out. You do a really wonderful job. So thank you for all of that. Thank you for your time today. I always learn a lot. Today was no exception. So.
Well, excellent. And I did too. And I really liked how you brought up some of the questions, and I really liked how you articulated your points, especially the philosophical stuff. Maybe we should do more of that,
That's fun.
or I should stay in my lane. Well, anyways, I will see everyone later, and thanks for the comments. Thanks, Aaron. And yeah, be sure to check out Aaron the Grower as he himself grows in prominence.
Thanks, brother.
Yeah. Have a good one.
You too.